Hello, and welcome to Introduction to Bioinformatics Analysis, a course taught by Phillip Richmond. This is Section 6, Data Organization. So we are going to open up our terminal, log into the Orsinus Westgrid compute server. And perfect. So in this presentation, this very short but sweet presentation, I'm going to talk a little bit about organization. Organization is very important, especially when your projects start to get bigger and bigger. It's very important to know where files came from, how files were processed, and documentation throughout the entire project is very key. Otherwise, by the end of the project, you'll be looking back at your results, which may be going into something like a publication, and you will have no idea where any of the files came from, and you might have to actually redo a lot of the analysis using file locations that you could actually reference on the internet for other people to replicate. So organization and documentation, both very important. There are many different organization styles, but in general, you can think of having a single project directory with subdirectories that can delineate your raw data, the data regarding genomes, perhaps the scripts that you use to process the data, and then a place for the processed data to live. So if this is a variant calling experiment, maybe you have within process subdirectories for your aligned data, your SAM files and your BAM files, and then perhaps you've got another directory for the variants that you may call. This is just one example of a hierarchy that we will be using in this project. So why don't we go ahead and take a look at the directory that we created from section 5A and 5B. So if we go to our global scratch WRC directory, we can go into the sandbox that we've been working on, and if I do an ls, you can see all the files are pretty much just stacked in a single directory without any organization. I have this one downloads directory, but that's about it. So what we're going to do is we're going to just make a few of these directories. I like to make directories with all caps sometimes so that I know the difference between a file and a directory. This is something that you can adopt, or you can use whatever works for you. So again, I have this raw data here, but we do not want to use spaces in our directory names. So I will make a directory called raw underscore data. Underscores are a good way to get around spaces. I'm also going to make a directory for genomes, for process, and for scripts. And so the first thing I'm going to do is I'm going to move the lambda virus FASTA file into genomes. I'm also going to move the lambda virus bowtie indexes into genomes, and again, I've done this once or twice. You can use the star or asterisk for referencing multiple files. So this says reference all files that start with this character sequence, and they could end with whatever. That's what the star means. It says, as long as you've got this, I don't care what you end with, that's what we want. And so by hitting double tab twice, you can see these are the files that are being referenced. So we're going to move all of these into genomes. And now if we do an ls, we can see we've already started to clean things up. I also have some S288C genomic information, and I can go ahead and put those into genomes as well. just to clean things up. Okay, the next thing we're going to want to move is we're going to want to move all of these bowtie2 BAM, SAM, sorted BAM. In the end, these are all the same file, and at the end of a project, you will probably end up deleting everything except for your final processed BAM. So you would delete the SAM and the BAM, but you would keep the sorted BAM and its associated index. But for now, let's just go ahead and move all of the lambda bowtie2 files. So we can hit tab twice. Yep, we want everybody to move into process. And then now we are almost all the way clean, and this is much more organized. It's kind of like having a desktop with 100 files on it. It gets very messy and it's very unappealing and very unorganized. And so this is the same idea. A bit of organization when it comes to our files will save us a headache in the long run. Lastly, we're going to move everything that ends in .fq, all our FASTQ files, into raw data, and we have our shell script that we can move into scripts. So now if we do an ls, we can see everything is much cleaner, and the second part of this organization is documenting where we got certain files. So it's good to have a README in every directory. This might be a little extreme, but it's definitely important to have a README where your raw data comes from and where your genome files come from. So I'm going to make a README here, and I will say these .fq files came from /global scratch wrc data raw on June 30th, 2016, my mother's birthday. And now we can Control X, Control C, yes, and just like that, now there is a README in this directory. You can call this whatever you want. README is just a bit of a standardized naming for, hey, README. So we can next go into genomes, and we can do the same thing here. Emacs readme.txt. The lambda virus.fa and associated bowtie2 indexes came from global scratch wrc genomes. Again, Control X, Control C, oops, Control X, Control C, yes. I haven't documented the S288C here. That's something that you probably would want to do inside the README as well. So again, now we can see we're much more organized, much cleaner, and we can then access our files much easier. That's all we have for this video. The only other thing to add on is that I like to have a very standardized naming scheme for files. So if you look at the process directory, I have essentially the name of the sample, the tool that it is mapped by, any additional processing steps like being sorted, and then the file type at the very end. So other examples for this, as we continue to process data, we're going to find that we might have a strain and perhaps a wild type, or a strain dash knockout, and as we have these files named in a very systematic manner, it makes it easier when we go to write scripts or parse files later on, or identify what a file actually means. If you just start calling things sample.cs SAM, then you won't know what tool it was mapped with. Things like that. That's all I have for section 1-6. Goodbye.